My name is Liran. I'm the CEO and one of the co-founders of BenchSci. I'm very excited to talk to you about what we do today. I'm going to cover the high-level problem that we're aiming to solve at BenchSci, and then I'm going to pass it to Casandra Mangru, who is our VP of Science and is a founding member in the company. Before we jump into what we do, a bit about the company. We are 130 people right now, full-time employees that work at BenchSci. We're growing to around 200 people this year. We've been pretty much doubling in size year over year. We work today with 15 of the top 20 pharmaceutical companies. We raised over $60 million, and we were Google's first investment in Canada. And with that, I'll start with our talk that is called 1 out of 10. So I would ask you to imagine working in an industry where you invest over $150 million in developing each of your products, spend 5 out of 7 years developing it, and that product has a chance of 1 out of 10 to actually work. Now imagine those products actually save lives. And that is the reality of the pharmaceutical industry. That is an industry that, as we can all see today, it shapes the health of our society. Yet 9 out of 10 medicine that enter clinical trials fail. 9 out of 10. The main activity to develop medicine and assess their success in clinical trial is through lab experiments, which are basically the first 7 years of the drug discovery process. And for each one drug that gets into clinical trials, there's over 7,000 preclinical experiments that are being done in the lab to better understand the disease and to assess whether or not that drug will be successful. But what the data shows is that only 20% of those experiments actually advance the study of those medicine. 80% of the work is actually not needed, and those are experiments that fail to advance the study of those assets, medicines, and targets. If we can fix that, if we can turn the 20% of experiments that do work to 100%, we can get novel medicine to patients years faster. And that is what we do at BenchSci. Over the last 5 years, we built an AI technology that can basically read biomedical data from scientific publications, patents, preprints, internal data from pharma, and so on, at the same level that a PhD scientist would. Today, we have decoded over 23 million experiments. And with that data, we help scientists from the top pharmaceutical companies in the world answer some of the most challenging questions they are facing today in preclinical R&D. Questions like, what are the most impactful experiments that we should be conducting on the bench? What are the most efficient way to perform those experiments? And how can we assess the reliability of the information that goes into clinical trials? And by doing that, we plan to bring medicine to patients 50% faster by 2025. I'm going to pass it to Casandra to share with you how do we do all of that.
Great. So how did we do that? So it's a very ambitious effort here. And I think one of the great things that we're super proud of is having an amazing team that came together to really help scientists with the main challenge that we faced, and one of the challenges that I faced at the bench, which is how do you get access to all of the millions and millions of databases and information that's out there that you need to make more informed decisions about the best reagents for your research and ultimately make better decisions about your experimental design. So essentially what we're doing here at BenchSci is decoding all of the different sources of information, extracting the key entities within these different scientific sources, extracting key entities of an experiment and putting that onto an intuitive interface that scientists can now leverage to actually get that information at their fingertips so they can do more successful experiments. So we started with pulling together a very diverse team of biomedical scientists, bioinformaticians, data engineers, machine learning engineers, and biomedical data analysts to leverage their respective expertise to extract the world's experimental knowledge. And we did it in a three-step approach. The first was to collect and curate all the different data sources where these experiments were located. That includes publications, third-party sources, any place where a scientist is doing an original experiment. Then we had to curate a reagent database. So So what are all the tools that scientists are using, like antibodies, as we heard earlier, to really study those proteins and understand how they're involved in disease, the mechanism of action of that disease, and the appropriate drugs to treat them. And then another challenge that we faced, and I'll touch on it in a little bit later, is around the bioinformatics. Protein and gene nomenclature is very ambiguous. There are many names, and we soon realized we had to pull together a large bioinformatics database and ontology to be able to capture that effectively. When we had all the different sources of data, we could then apply our machine learning to extract it, and we take a hybrid approach using both text-based NLP models as well as image vision-based models to extract data from these different sources. We contextualize that information by using tagger models to extract key experimental context. What are the cell lines, cell type, diseases, tissues that these scientists are doing these experiments in? This is important to note because not all tools work the same in every context. So having these additional parameters is very important for scientists when they're making their decision. That information is then QA'd by our team of internal scientists before it's made available on the platform. So in terms of the different data sources we have access to, we are looking at published information from both open and closed access publishers. We have partnerships to get access to full content. We're looking at preprint information like from bioRxiv and academic theses. We're also looking at third parties like Human Protein Atlas and Code that are testing and validating products. And they're doing experiments as well, like vendors as well, testing their products. And those are resources that scientists are referencing when they're trying to evaluate the quality of the products they're using. Then we had to build our reagent database. Now, this is no easy feat. One particular target can have millions of different options, tens of thousands for individual targets that scientists have to choose from. And all of this information is buried across disparate data sources, different sites, different repositories, and all of the information or specifications around each of these different reagent types is not standardized. So scientists often have to search individual terms. They find it very difficult to find different specifications. So one of the things that our team does is actually pull together all of that information. We create internal ontologies around all the specifications, standardize that to make it one of the cleanest and most searchable databases for reagents in the world. And we have antibodies, proteins, RNAi, CRISPR, cell lines, and we'll be adding animal model and PCR to that list very, very soon. Now, this is something that is has to be updated on a regular basis. New products are being created. We often find missing information, incorrect information in these databases. So our scientists are actually looking at that and making sure those gaps are filled so scientists can actually find that information effectively. As I mentioned earlier, one of the challenges we face is around nomenclature. So one particular protein target, using PD-L1 as an example, used for immune checkpoint therapy. It's also known as CD274, B7H1, and there are a variety of other names. Scientists can use any one of those names when they're citing that information in a paper, and vendors can use any one of those names as the target of their particular product. So to ensure that we're capturing that information effectively, we had to build our own ontology consisting to date of over 15 million different synonyms from over 170,000 different genes across hundreds of thousands of different species, and that includes microbial species, so that we can ensure that all of those different names, including hyphenating terms and acronyms, are all captured into and curated into one index term, so that that can ensure comprehensive data mining. But also when scientists are searching on our platform, they can be assured that all the data is together and complete. So now that we have all the different data sources, we can then apply our machine learning. So as I mentioned earlier, we're using both text-based and image-based machine learning, and that's contained within a very robust architecture. We've developed over 100 different models to analyze and extract relevant experimental entities. That includes detecting proteins, genes, the different reagents, the various applications that those reagents are being used for specific reagent types like RNAi and CRISPR. We also have dedicated sequence pipelines to extract that information as well. We use image-based or vision machine learning models to classify experimental figures, and I'll get into that in a little bit, and of course extracting the various experimental contexts. As many of you may know, your machine learning model is only as good as its training set. And when we started about 4 or 5 years ago, we realized that there were not any off-the-shelf training sets that we could leverage. What we were trying to do in extracting very specific experimental entities was very unique. So we spent the last 4 years, and this is a team we still have to date, labeling a ton of data, and that's in both text and images. Our team that's labeling this data are scientists that have domain expertise in the various areas of science research that we cover. Those scientists are not only labeling the data, but they're also helping in evaluating the outcomes of our machine learning. We've also developed internal tools so that we're able to do this at scale. So one of the first challenges that we faced, and we started with text-based machine learning, was analyzing scientific text. So scientific text, if anyone has read a scientific paper, is not like regular text. There are runoff sentences, punctuation is missing, protein targets are referenced in many different ways. And of course, linking that back to the outcome of the experiment, which is the figure, is very, very difficult. So we actually had to develop over 40 different models to be able to detect this information and create the relational associations effectively, and also help identify what is the output of that experiment. But we soon realized that text-based approaches were not enough. A lot of information was actually buried within the image. One of the things that we noticed a lot in reading these scientific papers that a scientist would often say, 10 protein targets were studied as indicated in the image above. And the images themselves are not simple. I have an example of one on the right-hand side here, where these images are often segmented into different panels, A, B, C, D, E. And within those panels, there are subpanels, and each of those subpanels represent individual experiments. So we created semantic segmentation models to detect panel bounds, both on the higher level and in the subpanel level. And we also have models that can classify individual panels into experiment applications. So we can tell that those middle four are IHC, we can identify graphs. Another thing that we had to make sure we extracted out of these images was, of course, the text. So we have text detection and OCR models that are able to detect embedded text, extract them no matter the orientation. So we have horizontal, vertical, we often see diagonal, extracting that text and then categorizing them into the various ontologies that we've built over the years to ensure that we're capturing information that's not mentioned by the scientist. We also have super resolution models to improve the quality of images, and to supplement those, we have synthetic images to ensure that we have large training sets. By identifying this data within images and text, we're also able to analyze a scientific paper as a whole, but in also identifying data within different sources independently, whether it be text alone or image alone. So now that we have all the entities, we have proteins, we have applications, we have reagents, we want to make sure that we're capturing the surrounding experimental context. Similar to the challenge we faced with bioinformatics, cell lines, cell types, tissues, diseases, species, all also have synonyms as well that we had to make sure that we developed ontologies for, especially diseases that have acronyms and many different names. All of this is curated by our team of scientists, and we're connecting that to all the core components of an experiment. As Liran mentioned earlier, we have over 23 million experiments for scientists to search from within our database. Now, before that data makes its way to the platform for scientists to see, we know that our scientific audience is very particular, and they need to ensure that they're seeing data that is going to drive their experiments forward. So to ensure the accuracy and integrity of our data, we pre-curate that data before it makes its way. And that is to ensure that there's a specific level of accuracy before scientists get access to it. We also have a constant feedback loop. We do data runs on our entire corpus of data on at least a monthly basis. We have feedback loops for our ML training. Any improvements to our algorithms are applied to the entire corpus of data. And we also have feedback from the tens of thousands of scientists that are leveraging the platform to guide the accuracy, expansion, and capabilities of the platform itself. That data within our database is powering the platform that scientists are leveraging today. They can search by protein targets, narrow down the results by specific experimental parameters to identify what other data supports the use of particular products in similar experimental contexts so they can be assured that they're using products that are going to work. And one of my favorite stories to tell about this is that we spoke to a group of scientists in our engagement with a particular client. They said, we had a project where we had to test at least 8 products to start to find the most appropriate product for a particular clone or protein epitope that worked in a specific cell line. And it took them 8 weeks to test those first set of products. They didn't find what they were looking for, and they took an additional 4 weeks just to find the one product that worked. So we put the platform to the test. They put in their clone ID, they filtered by their specific cell type, and in 30 seconds, we were able to find the one product that was validated in that particular cell type, and we could have saved them weeks and weeks of work. So that really shows the power of applying machine learning to scientific data, allowing scientists to navigate through the millions of data points that are out there that are there to really empower their decision-making around experimental design.
Interesting approach. Do you account for the quality of the research before you include this in the analysis?
That's a great question. I think looking at each of the experiments individually is really the key. And one of the things that differentiates our data is, of course, being able to capture things right down into the supplementary information. One of the things that we do on the data side is to look at, are there papers that have been retracted? Is there consistency across how experiments were done? And we can flag that to the scientists as they're evaluating the data itself. So being able to see the world's biological evidence together really allows you to see what has been done in the space. And one of the things that we want to do next is actually take a look at that and see how that compares across the different fields, because a scientist can be studying something in a very obscure space, but not realizing that that information is very valuable to another scientist. So pulling information together in this way allows us to see consistencies, inconsistencies, and also provide objective data for scientists to leverage.
Do you have any plans on engaging with the scientific community by sharing the ontologies that you've developed so that scientists can use them when they write papers?
I can answer that, Cass. The way we chose, so giving back is something that is really important for us here at BenchSci. And the way we chose to do that is by actually opening up a free version of our platform for academic scientists while focusing on pharmaceutical to monetize our platform so we can have more resources to continue and invest in our innovation and future product development. So we do actually have a free version that is open to scientists to help them accelerate their research and conduct more successful experiments.
The scientific community has become more aware of the replication crisis and the misuse of p-values in the scientific literature, especially medicine. How do these problems affect your algorithm, if at all?
Yeah, that's a great question. So pulling together, the first step is to standardize the information and understand where the experiments are happening. As I mentioned earlier, by pulling that together is the first step, and now we can go in and some of the advancements that we're working on is actually being able to detect specific entities within the image itself, protein bands, look at graphs, be able to compare across. So by pulling in the information, we cast the net, and now we can go in and really evaluate that on a level that can provide additional insight to the scientists as they're evaluating the data. So that is definitely a direction we're moving toward.